our world is constantly evolving. As time moves forward, things change, which is why it's important to stay informed. Throughout the year, there are dozens of professionals that share their expertise with the community through lectures sponsored by local government agencies and area not-for-profits. And each month, SLC TV will feature one of these visiting professors as they discuss the latest current events. So grab a notebook and pull up a chair, because the lecture hall is about to begin.
Good morning. Thank you for coming so early. It's I know how to get up early, but a lot of times it's nicer in the morning to get up early. So as she said, I am president of Fishing Oceanography, Inc., formerly owner and founder of Roffers Fishing Forecasting Service, originally in Miami, but then we moved to Melbourne 12 or 13 years ago. I sold that business last April, so I'm a part-time consultant for them. Started a new company in Melbourne Beach. So based on the work that I did at Roffers Fishing Forecasting Service in Melbourne over the last 20 years or so, some of the work we've done has been popularized with the media. Particularly, we did a lot of work with the oil spill, the Deep Horizon oil spill in the Gulf of Mexico, by providing daily oceanographic analyses indicating where the oil is likely to go. That reduced a lot of stress from people knowing that the oil was following certain currents, was headed for Key West, but an eddy came and then moved it offshore. And then other flooding events in the Indian River Lagoon, which I'm more focused now than I was before. Also been on radio and TV in relation to that. So my philosophy, as well as the company's philosophy, is green. So we really believe in sustainable consumption of natural resources. We believe in the right to fish, but we do believe in tag and release. So catch what you need for today, but leave the rest for tomorrow. Scientists need the information in terms of the migrations of fish, even though here we are in the year 2019, we still really need to know a lot about fish migrations. So any tagging that you can do or recommend to people, then that's great. So the other aspect of our philosophy is we know that habitat is critical, and water quality in particular is critical as well. And as we know from living around here, particularly with the Indian River Lagoon, how the changes in water quality varies very quickly, and the result is sometimes unfortunately devastating, as we've seen now in the Indian River Lagoon, where we have lost 80% of our seagrasses. Unbeknownst to people who think they just want to add more water, open up a new inlet, the solution to pollution is not dilution. Regardless how many times you flush that toilet, if you keep on putting poop in it, so to speak, you're going to need to keep flushing it, and more sewage and nutrients that go into the lagoon. You cannot possibly fix the problem by adding more and more water. So the solution to pollution is definitely not pollution. We believe in corporate and personal responsibility, so return the water to the system. This is a new concept, particularly for farmers and corporations. United States and most industrialized places of the world were developed by rivers. So the result is when you have excess water, you use the water in your power plant, whether or not it was a turning wheel or a power plant or in your manufacturing, you dumped it right back in the river and you said, that's gone. Started in the Hudson River and places up in Boston, Chesapeake Bay. Well, as time went on, we had more and more people, the population has increased tremendously. Now we can't flush that water, and the water's there. So I honestly believe that if you use the water, you should return it to the source in the same condition that you took it. So I also believe we need better science and more science funding. This is very important at this time. R&D is really what drives our economy in the United States. And regarding to fisheries, fishery independent data, that's rather than the catch and release, having scientists go out and collect data themselves, I think we need more of that. So just before we get into the Marine Biodiversity Network topic, which is the main topic today, the idea that everything is connected from the mainland. So even if you live 10, 20, 50 miles inland from here, whatever you put on your lawn goes into the canals, and eventually it runs down into the Indian River Lagoon and then out into the ocean. So out here, you can see it just flows right down, whether it's sewage that goes down or just flushing your toilets into septic. It leaks eventually offshore, and it affects not only the local waters, but as it's affected by the Gulf Stream, it then gets pulled offshore into the Gulf Stream. So putting trash, and people wonder how long does trash really last in the ocean? And this is a chart developed by Sea Grant up in MIT, but just curious, you look at a tin can, if it's dropped in the water, it lasts 50 years. A six-pack holder, we're talking 400 years. So if you see something in the water as you're walking down the beach, or if you're in a boat, please remove that from the system, because it's going to take an awful long time for it to recycle. Those of you who've been following the news the last couple of years, this is hard to read here, but the problem of plastic in the ocean is increasing. There are main gyres in the ocean, typically in some cases the size of Connecticut. That's the amount of plastic out floating around in area. You've been reading recently about a company in Hawaii, a non-governmental organization that has a technique to remove the plastic in the ocean. Unfortunately, the first round didn't work, but plastic is a huge problem, spreads all over. The other problems we have locally, as I implied, is the sewage, overdevelopment in areas. We love Fort Pierce and the surrounding communities the way they are. I'm personally originally from New York, went to University of Maryland in DC, and lived in Miami for 30 years, so I certainly am aware of big cities and development. I moved up here because I wanted to get away from that, and this is the kind of thing that the developers want to put in, but it actually reduces the habitat, and it hurts the ecosystem. So I am part of the Indian River Lagoon Council. I'm part of the Science and Technology, Education and Math Committee, so I'm heavily involved in water quality issues locally. And other aspects that are of immediate concern to me, of course, Lake Okeechobee, going into the canals instead of going to the Everglades. One, the Everglades are drying out. We need that water into the Everglades. Two, there's freshwater supplies going both towards the Naples area and Port St. Lucie. The organisms that live in the surrounding areas need some salinity to exist. If you're dumping this lake water into the surrounding, you're providing freshwater sources, which has actually essentially destroyed the ecosystem, both Port St. Lucie and the Naples area. Not only adding fresh water, but they've added Roundup to the situation, which people use to kill their grass. Also doesn't break down very quickly. It's very harmful for fish, insects, and birds. Up till recently, until the new governor was elected, they were using Roundup to spray around ponds, retention ponds. And while it keeps the weeds down, it also poisons the organism. So he's now put a stop to this, temporary stop, and I hope he continues. Roundup is a very bad thing. If you use it, I suggest you dispose of it properly. Do not use it again. So today's subject is on biodiversity. And it's a rather complex subject, and I'm so used to talking about biodiversity to other scientists, I wanted to first define what it is. So biodiversity describes a variety of plant and animal life that may exist in a particular area. It's the total of all living organisms, whether it's in a park, in a lake, a lagoon, or the ocean. So this is an important concept. So a marine biodiversity network, which is called NBON, is a systematic study of the life in the sea, from microbes to whales, from the surface all the way to the bottom, including the bottom communities. Those of you who have good memories or just remember what NBON or Marine Bond is, you can search that easily on the internet, and you'll realize that over the last several years, it's a growing initiative. People have studied different parts of the ecosystem in the past, called ecosystem scientists, but now there's a growing global initiative to form these regional networks to get a long-term data set so we can, between scientists and resource managers and end users such as yourself, whether you're bathers, fishers, or just enjoy the ecosystem. And the key is for existing programs is to work to integrate data from existing programs. And while it's hard to see here, basically going from the surface all the way to the bottom using a variety of equipment, drones, drogues, underwater submarines, and now something which I'll talk about later is something called eDNA, where basically anytime you go into the water or a fish is in the water, not only comes from the skin, but their urine and feces they produce or they release DNA. Now we have sampling techniques where you can go take samples of water and identify certain fish species or marine mammals just from the eDNA that's placed in the water. So the next time you go into the ocean and bathe and think that you're peeing in the ocean and no one's going to know about it, there could be a scientist sampling your water, and if you have your DNA sampled, we know who you are. Just kidding. But the idea of these marine biological diversity networks is to improve our understanding of marine ecosystem and the biodiversity. Ecosystem functions from ocean observations, particularly long-term ocean observations. Short-terms are good, but in order to understand changes, the variability over time, the longer the time scale that the observations are made, the more useful it is for science to understand the changes over time. So the benefits is just learning about biodiversity, about organism abundance, about system productivity, not just from the plants, but everything over the bacteria, the viruses, the bottom community, and the ecosystem services they provide. You say, okay, there's a fish, what does it provide to the community? Well, it cleans, if you go out in the tanks, you can see this cleaning some of the algae off the side of the tanks or on the leaves. Its feces go down to the bottom community, then you have crabs and other things living in the sand below that live on that. So it's a cycle, and we're trying to learn as it changes over time, how does the cycle change and the flow of energy change. So we're really monitoring ecosystem health in terms of the abundance of the fish or organisms. Are they declining? Are they increasing? Is it part of the natural change, or is it something being caused that's changing it? Whether it's pollution or fishing catches or droughts. So anyhow, this information from NBONs are being used by management, used to protect, to conserve, and utilize the resource. You know, if the populations of a certain fish species is abundant, no reason why we can't fish and use it to eat ourselves. So the other benefits, as we're doing today, is making the public aware of marine biodiversity networks. So it's not just a network that we have at University of South Florida, as I'll get into more later. There's the NASA and NOAA funded, and now it's the Navy involved funding these marine biodiversity networks. On the first round, there's networks in Florida, there's one in Monterey, and there's up in Alaska as well. So not only is it happening here, but it's happening around the world. As I've learned over just the last few years, scientists led by the UN in some cases, or by the national governments, are out there doing marine biological diversity observations as well. So this is part of the Group on Earth Observations. There's so many acronyms, still taking me a long time to learn these, so it's called GEOBON. I will try to avoid all these acronyms, but I'm just forced to using them sometimes. It's an international, and again, if you want to know more about GEOBON, just remember geobon.org, and you can find a lot of information about different things around the world. So what's the motivation? This is difficult to read and not perfectly clear with the lighting, but essentially, due to the increase in population around the world, there's a protein shortage. People need to eat. And if you have, if food is not a terrible lost commodity as it is in the developing nations, you still need to conserve and learn how the system works, so you know which parts you can use, you can replace, or you have to conserve. So coral reef, for example, is an ecosystem that's very important for the productivity of the oceans, as well as Florida, for example, and Hawaii, Australia. These are places where the productivity, as you can walk around here too and see the exhibits, the importance of coral reefs, are highly productive areas. As the ocean increases, the temperature increases, there's a lot of stress on the coral reefs, and we want to learn which reefs are actually benefiting from the temperature and which ones are being destroyed. Because the coral reef organisms die, then the surrounding ecosystem around them also suffers. So the United Nations has put together a series of sustainable development goals. That includes us as well as developing countries around the world. And there are 17 of them. And I'm not going to go into detail which ones are, or what each one is, but if you just look up Sustainable Development Goals, United Nations, you can spend the rest of your life probably reading about it. We're interested in number 14, life underneath the water. So each goal of the previous goals that I was showing you are interact with each other, and they're all connected, and each goal is important by itself. I would like to be involved in studying all aspects of these, whether it's poverty and whether it's climate actions or other aspects of it. I'm a marine biologist. I've been studying the oceans since the 1970s, so this is my main focus. But they're all interconnected. So again, this may be difficult to read, certainly is to me. Again, some of the other areas in the marine part and 14, we're interested in really 14.1, life below the sea, sustainable development, increased research capacity, and marine technology and biodiversity networks. So part of this, and I could do a whole, actually talk on this, and I spent a day in Nairobi last summer talking to the United Nations Environmental Program about this. But what they're trying to do, because everyone measures the ocean differently around the world, they're trying to define what are the essential ocean variables and what are the prescribed techniques, the special standard methods if you have background in chemistry. What will be the standard methods of taking temperature? What's the standard method of taking salinity, chlorophyll, water clarity, identifying the bacteria and viruses? These highly vary around the world, and sometimes the methods affect the results. So when you read in the newspaper or the magazines or the actual scientific article and try to compare one to the next, one of the things you want to really take a look at is, are they using the same techniques? Are the methods the same? You know, instruments may vary, but if they've been calibrated to a certain standard and it's been collected the same way, then you can compare studies. But sometimes the methods are so different that it appears, we're looking at ocean plankton. Well, how are you doing it? What's your standard? And this is something that's sort of obtuse, but the UN is trying to derive these variables and get governments actually to approve them. And this is going to be a 10 and 20-year process to just temperature, which is the most commonly measured thing in the ocean. After 5 years, they got it established. The next thing is salinity and chlorophyll. A lot of different techniques to doing this. I don't want to belabor the point, but just the ideas of using standard methods is becoming more and more important, so you can compare an ecosystem, let's say the Gulf of Mexico with the Gulf of Maine, with the Gulf of Aqaba or some other place where you're comparing fish and coral reef systems. Well, let's make sure we're comparing apples to apples and not apples to bananas or worms in some cases. So, so the indicators is something that, being that we can't measure everything all at once and all the abundance of fish, we develop these indicators. In fisheries, it's catch per unit of effort. It might be a video counting or laser counting. We're trying to develop these indicators, so rather than have to do a census of life around the entire world all the time to compare, we're developing indicator species of a coral reef community, maybe a series of them, or just indicator bacteria or viruses or crabs, birds. What is an indicator represents the health of that community? And it's important to align the biology with the physical mechanisms. This study, and something I've sort of failed to indicate, these are interdisciplinary. So someone like me who is a fisheries person works with people who are physical oceanographers and maybe satellite oceanographers, working with people working with DNA, people working with bacteria and viruses and crabs. Totally different curriculum and expertise. So we're all trying to work together, because what I see something in fisheries, someone may not understand in relation to the invertebrates or vice versa. So when we talk about fisheries, we're not just talking about the fish that are swimming that we see, but the ichthyoplankton, the eggs and larvae. What's affecting them? You can go just right here and look at the copepods here. The copepod community is so important when fish hatch from their eggs. The first species typically that most fish eat when they're hatching from their eggs are copepods. And there are different species of copepods, and there's different nutrients aspects of them, the fat content of the copepods that makes it important for the survival of the fish. So some of these I've already jumped ahead and gone ahead of myself, so I'm not going over this again. But by the year 2020, they really want to get certain of these methods, standardized methods, approved, and it's going. Not only it's being approached on a global basis, but each country, each economic zone is developing these for themselves. So eventually, we're going to take a standard method that has to be approved by the Bureau of Standards, and then probably by Congress if the way the UN plan is right now. And as you know, it's real difficult to get things through Congress for whatever reason possible. But anyhow, I've been working with the UN Environmental Program. It's centered in Nairobi, Kenya, but they have projects all over the world. And overall, what they try to do is they're not doing the research themselves, they're organizing it. So if they have a standard method that's developed, for argument's sake, let's look at chlorophyll, which is a plankton in the water. Once they have that method, then they go to different countries and work pilot programs to see how that works with the individual countries, so then it can be learned and used around the world. But this is part of the Intergovernmental Oceanographic Commission, IOC. You might be able to have heard of that before. And again, if you're interested in all about this, just search MBON or GeoBON on your search engines, and you'll find a lot of information. So the approach is collaborative. So I, as a fisheries person, is going to be working with satellite people. Going to work with bacteriologists, work with people who work with fish genetics or bacterial genetics. People who work with filters, look at different systematics, whether and learning the different species, and there are numerous species around the world. You look at them, oh, they look similar, but well, in terms of the tuna world, well, that could be a skipjack, that could be a bluefin, that could be a yellowfin. In terms of the different kinds of mullets we have around here, the menhaden mullets, other scaled sardines that look similar, but they're actually different, but they have similar ecosystem functions. So we're looking at it in space as well as time, looking at the abundance trends over time, and then the ability to produce maps, which, you know, when you look at a spreadsheet, it's like numbers on a sheet. Unless you're an accountant, bless them, because they love looking at numbers in a spreadsheet. I myself would prefer to look at a map, a nice colorful map, and seeing things. So what we're trying to do, and I've already mentioned it, I don't want to be redundant, we're talking about different pipelines, different categories that need to be linked. So we're looking at satellite data in situ, meaning collecting it by hand at the site, looking at the genetics and eDNA, looking at the biodiversity, and providing the information to a system called OBIS, which, if you're collecting data and you're a scientist, you say, well, I collect the data, I keep it categorized in my own way, in my own spreadsheet. Well, if you want people around the world to understand exactly how you collected that data and how it was sampled and how it was stored and how it was identified, then you have to put it in the format that Orbis uses. A little more complex, a little bit more work by scientists doing it, but if you want to be part of the system and be able to compare your work over time with other people around the world, you have to get into common databases, and Orbis is the big one. And then socioeconomic data is very important, because unlike right here in the group we have here, it's not an issue whether or not you have the money to go buy lunch or go buy breakfast. In other parts of the world, and then I'll go into later, if a fisherman doesn't catch fish that day, he may starve, his family may starve, his neighbors may starve. So very important of the value of the diversity of the species and how the ecosystem is working, and how it's providing services not only inside to other animals, but to humans as well. So I won't go into great detail about the indicators. As I did already, as I mentioned before, it's a metrics, it's a numerical scale, it's a lot of different things. It's an indicator of what's happening as a whole. We don't have the ability to count all the fish in the ocean as frequently as you need to do. So what we do most of the time is use the catch by commercial fishermen or recreational fishermen to understand the success rate or the failure rate over time. And if the catch rate goes up over time, one believes that the abundance of the species will go up. If the catch rates go down over time, one believes the indicator, the population of those particular species is going down. That's why we use different techniques to provide indicators, because we can't census the ocean every day. So there's a whole, again, you could spend days reading information about these bioindicators and the statistics used to develop them. I'm not going to go into the great detail. I did mention that the bottom line, it's going to be a lengthy process to get these approved, not only by the scientists in the same community. Heck, we can't even agree between the U.S., within the U.S., on how do you measure chlorophyll from satellites, which algorithms, which equations do you use to go from the light that's bounced off the Earth up into the satellite, how you derive chlorophyll. Very simple measurement. People will agree and argue for days, weeks, and months and years about what's the proper way of doing it. And never mind the United States, but overseas. Eventually, people are going to agree on, this might be my method that I'm going to use, but this is the world-accepted method, so you might end up using both. But ultimately, if it's going to go through this Sustainable Development Group plan, the goal is to get them accepted by the countries at the governmental level, at the highest levels possible. So I'm going to show you the underdeveloped part of the world first before I show you the more developed and more advanced systems. So as I showed you before, we're trying to sample the ocean from the surface to the bottom with the maximum amount of technology that's available, from satellites to eDNA, filtering the water, digging out the bottom, using microscopes, I mean labs like we have here. These, as you may not be impressed by just simple microscopes and looking at copepods or seeing tanks and looking at how the fish interact, that's not very common around the world. Now if you look at a country like Tanzania, and I was fortunate to be able to spend 4 or 5 months last year because my wife had a World Bank project in Tanzania. If you look at the ships that they use for fishing, I mean, here's a boat that's probably 20 feet with about 20 people in it. This is a fishing boat with a sail. There's fishing boats over there using as rowboats. People over in Tanzania and undeveloped parts of the world, including South America and Asia, do not have the mechanism, do not have the money to buy the fuel, the motors, or the electronics to go out and find fish, catch fish like we do here. So this is essentially the fleet here. If the bulb was brighter in the back, which it's not a big deal. I think you can still see very primitive style boats. I mean, some of these boats, you probably saw pictures back of Cleopatra in Egypt. This is the same style, and it's been successful for thousands of years, and they're using it here. I mean, this is a mode of transportation. Bicycles. Yes, they have trucks. Yes, they have automobiles. But if you can't afford it, how do you get your food to the market? And bicycles and things like this, and walking is a very common practice there. So if you have a fish, how do you get it to the market if you don't have ice or can't afford ice, or there are no ice machines? So most of the fisheries in Tanzania and in developing worlds is a very local based market. You catch it, you immediately go right to the market, and it's sold today. By the next day, it's probably perishable, and people will start either cooking it or salting it to preserve it for the next couple of days. But refrigeration is not very common. So is the NBON system, with all the sophisticated measurements that we prescribe for a developed world, is it relevant for Africa? I mean, as I just showed you, the artisanal fisheries. And they have limited resources at both the university and government level. I mean, they have plenty of humans, and they have very little actually sophisticated equipment and vessels. For example, in Tanzania, there's one big boat in the Navy. That's it, one. They don't have a Coast Guard, they don't have anything else. There's no large research vessels like we have here, like the Smithsonian uses around the world. They don't have heavily industrialized fisheries that other places might have to use that as a basis. So the industrialized fisheries in places like South Africa, yes, is more sophisticated. Or Morocco, yes. Or Egypt, they have the capabilities. But other countries around Africa and South America, the developing world, don't have the resources to do it. So what is really needed to help these people? What can be done? What can they do to contribute to this and help manage their own? What can be realistically achieved? Well, essentially surface measurements. They really don't have equipment to go down. They don't have salinity measurements or probes to put down in the ocean. They don't have the vessels to go there and to repeat. They just don't have the money to buy them or to calibrate them. So subsurface, in some cases, maybe from divers and observations from divers, but some resource abundance estimates from looking at catch statistics, yes, but not as sophisticated as they have in the developed world. Routine measurements in time and space. Again, if you don't have a boat to go out and take measurements, that you have to borrow a boat or go with a fishing vessel to do it, it's very difficult to do routine measurements in time and space. However, in Tanzania, I was very surprised to see, and I worked with the Tanzanian Fisheries Research Institute for the period of time I was there. I would go once or twice a week. They had a new, what they called an e-satellite system. And basically, it was a series of 3 computers which they received a satellite data stream from the European Space Agency. I was really shocked. Considering the kinds of things I showed you before, I went to the Fisheries Research Institute, and they seem to be reasonable. They don't have the more modern computers that you and I have. It's not air conditioning except in the 1 or 2 rooms where they have the computers. But these people were able to, with the satellite data they had and the little help from me, are able to look at changes in chlorophyll over time and change in temperature over time to understand the changes of heating and how it affects the chlorophyll. And I keep on mentioning chlorophyll and productivity. Well, chlorophyll is the basic group that's measured because we're looking at abundance of plankton. And you say, oh, okay, what's the big deal about plankton? Well, most of the fish species are linked to fish that eat the plankton. The mullet eat the plankton, the menhian eat the plankton. If you don't have things that eat the plankton, the grazers, then the top level predators don't have anything to eat at all. So chlorophyll is a very good indicator, and we're looking at cycles in relation to chlorophyll and temperature, but without going into this too long, they were able to produce graphs like this, not only temperature and salinity, and use satellite data, just with a little bit of help and a little bit of training. So the bottom line, they can do a lot. The scientists, I was surprised to see that had a bunch of PhDs and masters and bachelors of science that initially are educated inside of Tanzania, but they're all required to take internship in Europe. So these researchers, the people I work with, where you think, okay, they're in very undeveloped countries, they have seen sophisticated techniques, they've been trained properly, and these people can really do a good job given the money, given the resources, so they can buy the computers and buy the equipment to do their work. So the bottom line is, yes, they can do surface and probably some subsurface, but that's the best they can do right now. And again, as I mentioned, these other developed countries in Africa can do a whole lot more. So now let's look at more advanced systems. And I didn't pay attention to the time. Let me just see where I am in terms of time. Okay. So the US and South American Biodiversity Network, using satellites, we're going to look at different parts of the world that have set up these observation networks. And while you don't see Chile or Peru, their country and their fisheries people are doing plenty of research. It's just they're not part of, right now, the GeoBond, which again is a developing concept. In the US, BOND and the Florida Keys, the Flower Garden Banks, which is part of the projects that I'm involved in directly, Santa Barbara Channel, Monterey Bay, and also up in Alaska. So in the Florida Keys, there's a great effort to collect eDNA, to do coral reef fisheries abundance estimates. We're collaborating with a lot of different scientists. And important, I haven't mentioned the word yet, is collaboration. One person, one institution cannot do all the work possible. You have to work with other people with expertise who have specializations in those things. So you work with other institutions. So you have a group at the University of Miami that does the fish counts. We have people at the University of South Florida that may do the computer work. You have someone originally from Woods Hole is now up in Oregon doing seascapes and eDNA. So this is the map of the different stations we have in the Keys. So the idea is not only do we produce a lot of numbers, but what are the numbers that are in terms of utility for the common man and management? We produce these infographics showing the ecosystem, what's part of the ecosystem, the bigger fish and smaller fish. And if you get on the website, which we're not going to do here, again, the NBON, University of South Florida, all these are links to different aspects, different counts. For example, reef fish richness. Well, how does that change over time? You look at 1999, since this work began, and other measurements we go much longer, back to the 80s and even 70s. And if you're looking at different oxygen isotopes in coral reef back thousands of years. But if we just look at fish reef richness, you can see how it changes over time. Some species go up and then flatten out, some go up and down, and we try to understand, the purpose of this is understand what causes these fluctuations in fish richness, the number of different species on a coral reef head. And that's an indicator of health. Is it environmentally induced or is it man-made induced? Is it from sewage? And a lot of the coral reef disease we have in the Keys is due to sewage. Others is due to temperature stresses. So we talked briefly, or I mentioned briefly, about environmental DNA. Again, fish or other organisms produce it, goes in the water, and you can separate different species, identify different species. We haven't been able to get into abundance yet, but numbers of species, which is one indicator of richness or species health or reef health, can be done with eDNA. It's a new technology, and you'll see and hear more about this over time. Just too dark in a room to show you, but the Monterey Bay is looking at in the bay and the effect of the offshore changes and the changes of temperature, salinity, oxygen on different species, their abundance and their movements. Looking in that case for forage species. So one of the projects that I've been involved in for a long time, being I'm a satellite oceanographer, is using satellites to look at daily, weekly, monthly, and seasonal climatologies in temperature and dissolved organic material from chlorophyll, different other measurements that we see around the world. And this is important. The satellites are great. They have the ability to sample the Earth in 90 minutes. So it takes 15 minutes to go from the North Pole to the equator with some of these satellites. It goes over and over and over again. What you see on TV for the weatherman, he's using typically one satellite, a geostationary satellite. We're using 18 different satellites to get these measurements on a daily basis around the world. And places that you couldn't easily sample. I mean, not in the middle of the ocean or some of the countries that don't have the capability to do it. Satellites basically covering the entire Earth all the time when you're awake or we're sleeping. So another aspect of the NBON, which is getting more and more important, is the seascapes. By taking these variety of measurements, temperature, chlorophyll, turbidity, we can identify preferred habitats from different species. So if a species likes, for argument's sake, 80 degrees with intermediate turbidity or transparency of the water, if you can find where that is on the map, then you outline in colors where that zone might be, where they are, and you look at that, how it shifts over time. And you'd be amazed that if it's a cold winter, some of these may shift to the south. If it's a warm winter, they'll either stay where they are or they may move further north. The migration of fish and organisms is very much controlled by temperature and ocean productivity. And using these skiscapes allows us to map basically the whole Earth using the satellites to see how these habitats move. As a fisherman, this is important because, okay, you're going out and catching kingfish, let's say off of Fort Pierce, king mackerel, fishing is great. You figure, okay, next month it's going to be great as well. Next month there's a whole series of cold fronts that come down, similar to what we have now. Now you go out at Fort Pierce and you can't find king mackerel. Well, did you catch them all? In reality, that habitat has moved down to Florida and the Keys. So if you have a small boat here and you can't get down there, for you, it's a bust. You don't have that. Your catch point of effort declines. You have to find another species to fish or move someplace else. The other benefit to these seascapes is that if you're a fisherman and you know the habitat's not in your zone anymore, you're not going to waste the time and money to be fishing for that species in the area that you normally do. So you have to change your gear, change the species that you're looking at. So that's the importance of seascapes. So I mentioned University of South Florida and other organizations. The Smithsonian is heavily involved in biological observation networks. There's a big ocean microbiome project in which it's a citizen science project where they have sailboats collecting water samples for them around the world. And the problem is data. Being able to get data. Take that sample. Maybe not be able to analyze it, but take that sample wherever you are. You're walking down the beach, grab a sample of water. The health department doesn't have enough people to sample. The state doesn't have enough people to sample. The feds don't have enough people to sample. So they need, as time's going on, trained citizens can very easily go measure sea surface temperature, measure water clarity, and provide the information by email or even a letter in some cases. But anyhow, they're measuring plankton, salinity, and temperature around the world. It's a very popular, and it says a long-term benefit is looking at ocean health over time. They are looking at things like ocean acidification, algae blooms, marine plastics, as we mentioned before, ecosystem dynamics, the change in heating and cooling, productivity as it shifts, and what's causing it. And the great driver today is climate change or environmental variability from things like El Nino, which is a large oceanographic, weather oceanographic system that affects the ocean climate. The other larger project is by the Tannenbaum Marine Observation Network, which again is the Smithsonian. It's called, I think I have the acronym here somewhere, TMON. So basically their focus is on the coastal zone, not the wide, large ocean. The mangroves community is so important. They look, there's a mangrove, big deal, it has roots in the water. The roots provide nursery protection for organisms. They provide bacteria that develops their own ecosystem in itself, what allows juvenile fish to grow and be protected before they move out and then grow up and become either a food source themselves or eaten by other fish to be eaten. So they're focusing on the coastal zone, but it's a global open source data repository which they have, and they're developing it and then contributing it to the World GeoBond. So they have 6 major projects. This is the TMON, the Smithsonian's. They have one in the Chesapeake Bay on crabs, marine biofouling. They have one in looking at corals in Panama. Coral biodiversity, not just in Panama, in other areas. They're looking at underwater meadows, which is seagrasses. So important. We lost 80% of our seagrass in the IRL. Well, fish need to eat the algae off those seagrass. The mollusks, the little cones and whelks that eat clean the seagrass is important. If those disappear, then the fish have nothing to eat, and they disappear too. So if you're a fisherman and you've been fishing in the lagoon, Indian River Lagoon for the last 10 years, you've noticed a decline in your catch. It's not because you don't have the ability to catch fish anymore, it's that the abundance of the fish has really dropped tremendously in the Indian River Lagoon. The good news is, in the last year, up to recently, we've had a drought. So the water is clearing up, and any seagrass that's left is starting to grow back, and we have a long way to go. It's probably going to involve replanting seagrasses in the community. So a technology transfer like I'm doing today, but the UN has a program, GeoBond has a program called Bond in a Box, where they provide all the equipment, all the methods in a box, and literally it's the size of a crate. It can be shipped to any country, any place around the world, and you have a startup kit to doing a marine biological observation biodiversity network. So it's called Bond in a Box, pretty cool, and it's really, really a nice setup. So we talked a little bit about citizen science previously, and this is something that everyone in the room can do. If you're motivated to help science, to help the Smithsonian, help anybody, water sampling, taking samples, ask the Smithsonian, whether it's this side of the street or the other side of the street, where do you need samples taken in the lagoon? Well, again, as I said before, the county and the health department can't sample everywhere. Typically, they'll sample on one side. Well, what about the other? Well, we can't get there. Well, if you have a boat, if you have a kayak, or you wade, you can go places and take samples that they can't do. You can repeat samples. Basic water sampling in a bottle, take it to them. They can take salinity. Often you'll take temperature from it, and they may even get chlorophyll measurements. You can do easy censuses of marine life. So if you go to a certain pier every day, count the number of porpoises you see. If you go there between 9 and 10 for coffee or breakfast, the same place, How many porpoises do you see today? Write it down. You also can write down the weather conditions. That's helpful too. You go back the next day, the same. You do the same and same. Even if you just do it weekly. All right, we've gone every Tuesday, we're eating at the pier. What are you seeing there? Those numbers over time, put together with other people taking similar measurements, will give information that's useful for scientists, for the conservation. Birds, number of nests. You know, you go to an area, you walk in the park, how many osprey nests do you see? Do they have young inside? Well, that's important information. The abundance of ospreys. If there's no mullet around, no food sources, the bird population will suffer. Pelicans as well. How many white pelicans do you see at a certain park? Count the number, write it down. What's the wind condition? What's the temperature? What's the time of day? Over time, you might see a pattern. It may be affected by the temperature, the air temperature, and the wind patterns. That's just useful information. Watercolor. Well, there's something called a Secchi disc, and we don't have one in the room, but basically it's a little circle, like a paper plate, painted in black and white triangles. As you measure it down, the distance where you see it and then where you don't see it is related to the visibility and the clarity of the water. Secchi disc measurements are very important to calibrate the satellite data for looking at plankton and turbidity. So something as easy as a little Secchi disc on a rope, you put it down, you take the measurement, and you provide that information, if it's once a week, to whoever you're working with as part of your citizen science program. There are iPhone applications where you can take watercolor pictures and send that to people, because that also gives you a measurement of water clarity and the color of the water, which is very important for calibrating the satellites. Plastic debris counting. You go, oh, well, there's debris everywhere. But as you're picking it up, if you're a good citizen, you're picking up trash as you're walking up the beach, count the number of pieces you're picking up. Is it affected by wind? Record that information. What types of it? What's the size? Is there colors? Bottle caps? What's more predominant when you go to the beach now? Is it bottle caps? Is it water bottles? Is it miscellaneous plastic? Is it stuff that comes from the cruise ships? Is it coming from Cuba? To me, as I walk the beach, I'm always interested to see. I open and see if there's an indicator on the label where it's made. After storms, tropical storms and hurricanes, you see stuff made in Mexico, made in Cuba. To me, that's exciting. How far has this gone? If you collect what they call sea beans, which I didn't mention here, these are seeds that travel from South America, even from Africa, that get washed up on your beach. You look, oh, this is an odd-shaped thing. It looks like a hamburger. Well, it turns out that's a hamburger seed. It's part of a sea bean, and that's important, is where it's coming from. This helps oceanographers start knowing, even though we're looking from satellites, what's really being transported in the currents. So, diving with a purpose. If you go if you snorkel, count the number of fish, the different types of fish, record it. You go the following week, the following month, what's changed? Record it, send that information in. As minor as you think that is, oh, everyone dives, everyone knows this. Well, if you take the number down and you write it, that goes into data and actually can be analyzed and be used, as opposed to, yeah, we saw 3 clownfish, pretty fish, this. Well, that's helpful for someone who's studying biodiversity and changes and understanding the coral reef health. Invasive species. If you're going there and you don't want to count everything that you see, count the number of lionfish that you see. That's important. Lionfish are invasive species. Don't touch them, but just identify them and say, oh, that's a lionfish. Geez, I saw 10 today. I saw 15. Oh, there's hardly any here. I happened to notice a grouper eating a lionfish. That's a great observation. That should be informed to people. And then coastal flooding, sea level rise. You know, I don't know if you've noticed it, if you go to the piers and different properties during full moons, certain times of the year, certain neighborhoods are starting to flood now. The mapping of the flooding is not well known and not well described. You read in the newspaper, South Beach. Yes, South Miami Beach is flooding. Every full moon, it's flooding. Up here, we haven't seen the kinds of rises that they have there, but that may change. Hampton Bays and in the Chesapeake, Virginia Beach is suffering from sea level rise. Lower Manhattan, sea level rise. So people are talking about arming beaches, putting dikes in, all sorts of stuff, but we need to have the counts, and that can easily be done. Oh, I have a house on the beach. Boy, the water seems to be coming more now to my dune crossover more frequently than it did before. But if you just sort of, well, how many times did I see it last month? Was it 3? Was it 10? If you take a number down and you write it and share it, it's a record. So I think I'm pretty much running out of time now. And ask people here at the Smithsonian, at the aquarium, How do we collaborate? What kind of projects can you get involved in? And with that, I'll end it.